Record. Um, and then share my screen. Okay, meet one more person. So thank you everyone for coming in today. First, I need to apologize that I forgot to send the reminder email, but I'm glad that you've shown up today. So today we are going to talk about the RC typing workflow. So, which actually is the main topic for this training that was also developed by JIDC UK. So getting into it. So RC typing workflow is a pipeline for the generation of phylogenetic trees based on ribosomal proteins. So before we go ahead, I'm just going to send you a couple of links for your own interests, which you might have a look at later. So the first one is the GitHub link for the original SnakeMake developed workflow. And then the second one is the Whittle version of that that was developed by Theiagen. So, you could just have a look at these if you want to go through the workflow itself. And then I'll just post there as well the link to some of the documentation for the workflow as well. Okay, coming back to my slides here. So you have the main workflow in blue. That color could be colorblind. And then you have two separate sub workflows. So just to let you know that the Whittle implementation of this mainly actually at the moment is just on the main workflow. So the Whittle documentation, or rather the workflow that you'll find on Terra, does not have the sub workflow that can download sequences from NCBI. But we do have I think we do have this bit with the Diamond database, small database wrapped up in the Docker image as well. So basically, this shows the step-by-step working of the workflow. So the next slide, I just explained in general what this entails. So as input, you need assemblies of your samples. So these assembled genomes as typically would be like GenBank files. So they should be well annotated because the workflow expect that you have .gbk or .gbf files that are not just the faster version or the config assemblies, but also annotated by something like Proca, for example, which is part of TheaProc. So if you did your assembly using TheaProc, then you do get a GenBank assembly, which you can use with this workflow. So once you input your assembled genomes into the workflow, the pipeline identifies and extracts sequences of 16 conserved ribosomal proteins from the input data. So this is mostly done again using the metadata information that, when you do your annotation, is typically, identifies the protein quite quickly. And then you have sequence concatenation. So what this does is you have your assembly, you extract the 16 sequences, and then you concatenate them in a sequential manner to make one sequence for each sample, okay? And then with each sample now, concatenated into a single sequence, you do a multiple sequence alignment where you align homologous regions for the different samples, obviously for the 16 conserved proteins. And then with that alignment, you generate a phylogenetic tree, yeah, to identify the evolutionary relationships between the different samples. So the key takeaway here is you need a GenBank file that is annotated. You need references that consist of 16 ribosomal proteins, and these are conserved through the different or rather the bacterial kingdom. And then the workflow concatenates these sets of sequences, does a multiple sequence alignment, and then the phylogenetic tree construction. So going back to this, I mean, if there are missing sequences, so if, let's say, for example, your assembly does not have all the 16 ribosomal protein, then we have a sub-workflow that would blast these sequences against the Diamond database to try and identify these. And this can happen, for example, if your annotation couldn't identify the particular ribosomal protein. And if that works out, then, you know, the sequence is then concatenated to the previously concatenated sequences that were missing some of those proteins. Yes, Rotimi.
Thank you, Jim. Well done. So I'm not sure where the issue of a reference comes into this. I don't have a June annotated COS, and I want to specify the reference
Rotimi, you're breaking up.
Oh my goodness, okay.
You said something to do with... Mm-hmm. Yes.
So, I mean, I'm not sure the issue of reference comes in here as to before we concatenate before we extract the soma and
unfortunately, you're cutting out. I can't quite get. Are you asking about are you asking about like the reference proteins? Are you asking about the assembly themselves? Okay, I can see the reference. Okay. All right, so I'll talk about the reference in a few slides, so I'm glad you've asked. Okay, so why ribosomal proteins? So let's start here first. So generating these trees based on the ribosomal proteins is crucial for several reasons. So remember, we are working with highly conserved ribosomal proteins across different organisms. Actually, I could get you... so if you looked at the... let me just get that. I think it should be... I'm trying to get the actual paper where this is detailed. It should be this. Yeah. So, so the reference, good question. So if I can just go back before. So the reference protein actually does come in because, okay, think of it this way. You have your sequence and then you have reference. So you use the reference protein as some way of blasting or identifying the complementary sequences from your particular set of assemblies. I don't know if that makes sense, Rotin.
Yeah, thank you, James.
Okay. Yeah, and the paper that I've just sent to you gives you some idea about how they identify these 16 conserved ribosomal proteins. So obviously, if you have, a set of proteins that are conserved across different genus, you have conservation, then it makes it quite easy to do comparison even within or between pathogen within the same genus or between different species. So it's obviously a reliable marker for determining taxonomic placement. And of course, we use this also for epidemiological surveillance. We can be able to track the evolution of these pathogens and also monitor outbreaks. So the 16 ribosomal proteins, as I said, are universally conserved and provide a stable and reliable basis for constructing these phylogenetic trees. The list of these 16 ribosomal proteins are listed here. However, note that the reference files that we're using for this training are from Staphylococcus species, but I did talk to one of the developers of RC typing, Lisa Crossman, who's been able to send me a couple of additional references for, just get that list. So we do have references for Pseudomonas. We do have references for Campylobacter, as well as references for E. coli that she has pre-prepared. So if you needed to run this, particularly on those 3 additional pathogens, then it should be possible. Currently, I'm testing this on some Vibrio references as well, just to see if I can be able to make the reference proteins myself as well. And then be able to also show you how to do that for your specific pathogen. Hopefully that works out as it's currently running, and we'll know in a few if that did work or not. So basically what is expected of this ribosomal protein is that the naming has to follow a specific convention. So you have the L or the S. So depending on the S should be the subunit and the L should be the large subunit, or whether it's large or small. And so this has to prefix with the L2, L3, whatever that number for the subunit is, as well as the protein itself. It doesn't matter what you have, after that, but as long as, it has to start in this manner. In some cases, you might have the nomenclature flipped a bit, like in some annotations, you might find that it starts with, for example, LPLB underscore L2 instead. But so that you don't have any problems with the workflow, then, it should be named in this manner. So it's a long list with different functions. So I'm not going to dwell into what each does, but as part of the slides, I have a description of what each protein subunit is. So you could look into those a bit later, of course, together with the paper that I just sent. Where did my mouse go? Okay. So are there any questions up to this point? Just on the description of the workflow before we get into looking at the workspace data elements, as well as the data table, and eventually being able to run the workflow itself. Any questions at this point? All right. Okay, cool. So now we want to talk about workspace data elements. So we did last week or the week before that talk about how to upload your data and how to run it for a workflow. One thing we didn't talk about is the workspace data elements. So if I just switch over to Terra here really quickly before I get back to the slides. Oh, so that is loading. I'll just quickly sign in. Okay, I'll just reload this and hopefully this time it signs in much faster. Okay, that works. So if you look at your Terra workspace towards the bottom left, you'll see a section called Other Data, and you'll see Workspace Data. So we just want to talk about what this is. Okay. So going back to my slides here. So this is data that is generally shared for use in any workflow within your specific workspace. So this could be reference genomes that you use across different workflows. They could be like bed files that have these primer coordinates. So if you've run SARS-CoV-2 analysis, you'll be familiar with Attic v3, Attic v4, the Midnight Protocol reference files, sorry, as well as it could be like a Docker file or an image for the latest software for a given tool within a workflow. You know, in general, you want these to be like most up-to-date files for analysis within that particular workspace. So assume that within the same workspace, you do undertake analysis for different pathogens. So you want to have a reference. Again, remembering that, in public health, it's often good practice to have standardized protocols. So you want to have the same reference, for example, for Vibrio. If you're running Vibrio analysis, you want to have the same reference for Klebsiella or even for SARS-CoV-2, just to ensure, you can compare results across different runs. So this is where you would store such references within the workspace data elements. And as I showed you, the workspace data looks something like this. So within the data tab, and then at the bottom left, under other data, you'll see the workspace data there. So you have different or three columns within the workspace data. So you have the value. So this is either the file or like a string that represents the data element. And then you have the key that identifies that specific value. So in this case, for example, we have Attic v3 primer bed that identifies this specific file. And we have Attic v4 one primer bed that identifies that specific file. So you'll notice these ones in blue here are actual files, whereas these ones that don't have the bluish color are just strings, okay? So obviously this key value pair is quite important because you identify the value using the key within the workflow. So what you link on what you give as input in the workflow is actually this key column here. We have a description column that is not mandatory you put something, but obviously it's quite useful to describe what each data element is. So, sorry. When using this workspace data element within a workflow, we use the following structure. So you start workspace dot key. So you'll remember that, when you were setting up the sample level workflows, even in Theiagen, we started with this dot something. So in this case, this, as we said, identifies data or a particular column within the data table from which you're getting your samples. And then now we are using the notation workspace dot to identify a workspace data element. So going back here, if we wanted to input like this Attic v3 primer bed file, then what we would do is to say workspace dot Attic v3 primer bed. So that would identify that, we want to input this value, and we identify it with that specific key. All right. Any questions? I can quickly switch here. Probably this can come up like at the moment, you can see that in my workspace, I have two workspace data elements. There's this Staphylococcus zoonosis, and we also have Vibrio cholera references. So these are the ones that I'm currently testing. So adding a new workspace data element is actually quite easy. So if it is, let's say, just a string, which means it's not a file, then you just come up here, edit, and then you go to add variable. So you can literally say, let's say, Klebsiella, and then let's assume that it's a string. In this case, you have to do the double quotation. So let's say v5, let's say it's something to do with a version in this case, and by version. And if we save it, it should be saved. So then you can call that particular workspace data element into your workspace in that case. In this case, I'm just going to delete this because we don't need that specific one. But what happens if you need to actually add a workspace data element that's supposed to be a file, let's say this star file? So the way to do it is you just come to your import data, upload TSV. No, open data uploader. Sorry about that. I would choose the workspace. So in my case, it's that specific one. You can either put it in an existing collection or you can create a new collection. So, it doesn't really matter. Again, depending on how you want to organize your things. So let's say here you have your data. You can literally create a new collection that you call references if you want. So let's create one. And once you've created these, I can pick a file to upload. Let's see. I mean, we can upload any file really. At this point, it doesn't really matter. So once I've uploaded this file, unlike previously where we needed to upload a TSV file so that it creates a data table, I don't need to do that. All I need is to get a link to this file. So if I hover my mouse over this file, you'll see that, it gives me a link at the bottom left, or rather I could just also click on it. And it gives you a few more details, like if you want to copy this, it's giving you the path. But I don't want to do that. So the best way to do this is when you bring your mouse to this clipboard here, it will tell you copy the file URL to clipboard. So I'll just click on that and that will be copied. And then I can just go back to my workspace and to the data column and to the workspace data. Again, I'll just go through the same process, add a variable. So let's say I can do the same Klebsiella. In this case, I'm going to change this to be, well, still a string, but in this case, I am putting in a string that is a file. And here I can just again call it reference. And if I save it there, you'll see it generates a link to this file. And in this case, you've now created your workspace data element called Klebsiella with the value that is a file called data_2_fetch.tsv. All right, I'll just get rid of that again, because you don't need it at this point. But that gives you an idea how to have these workspace data elements. So again, remember, this is not for input data that you want to analyze within the workflow. So that is normally residing within these data tables, but this is more of like some files or inputs that you need to use across different workflows within the same workspace. Okay, are there any questions? All right. We'll see shortly how to actually input the workspace data elements once we get to launching the RC typing workflow. The next thing that we want to talk about are the set level data tables, because this is also important for RC typing workflow. So there are two kinds of data tables on Terra. We have a sample level as well as a set level data table. So each sample level data table can have an associated set level data table. As you've noticed, if you ran Theiagen, you started with training data, and then you had another data table created with a suffix underscore set. So for example, here in this slide, if you had a sample data table called Illumina PE specimen, once you run a workflow on that table, you'll see an Illumina PE specimen underscore set data table created. So unlike a sample level data table where each row represents a single sample, on the set level data table, each row represents a set or a group of samples. So these samples themselves are actually on the sample level data table, okay? And then the set level data table gathers the information about the set of samples together. Now, sets of samples are obviously useful for comparing samples and combining data together. And, you know, as we did the introduction, the RC typing workflow generates a phylogenetic tree based on these 16 conserved ribosomal proteins. So obviously you need to aggregate samples, different samples, to be able to undertake that phylogenetic analysis. Okay, so here, if you look at this workspace here on the data tab, so you have the sample level data table here, and then the set level data table here with the underscore set. And of course, similar to, you know, the sample level data tables, you have sort of like this database nomenclature where, you know, you have entities, which are the rows, and then you have the columns, which are the attributes. So data within each column of the set level data table either represents an attribute about multiple samples or could be the results from a set level workflow. Okay, so to summarize this, a sample level workflows take information from sample level data tables. So Theiagen, for example, is a sample level workflow. It takes data from each sample and processes it independently. And then the results are populated at the sample level within that table. The set level workflows take in a set of samples, returns the results to a set level, like where each row represents a set of data, and will return the results to that particular row that represents a set of samples. So the Theiagen Illumina PE workflow that we ran last week is a sample level workflow that takes in each sample and then does that bacterial characterization and assembly. RC typing that we are going to launch this week generates a phylogenetic tree based on ribosomal proteins from multiple samples. And if I can just flip back here, so just going to our training data, which, you know, we were undertaking assembly, QC, as well as characterization, you'll see that, we have 42 samples. Each row represents a single independent sample. We did run Theiagen, and you can see that, we also ran SRF-Fetch. SRF-Fetch, we ran it on 40 samples. So you see for each of those workflow runs, it creates a row that represents that set of samples. In this case, it doesn't mean that we ran the workflow on the set, but rather the workflow created this row to represent, the set of samples that were run for that particular workflow or workflow run. And here you can see that I did run Theiagen twice. In each of those cases, you can see that, we had these 42 samples created there. I also ran another workflow called zip column content, and you can see. So for zip column content, so this is a set level workflow. So whereas SRF-Fetch and Theiagen populated the results back to the sample level workflow, this zip column content, which essentially what it does is, think of it like, in this case, I needed to download all the GenBank files from my training data table. So what it did is take all the files from each sample and then zip them into one file. Then I can be able to download that instead of downloading each sample independently. And once I download that, you can see the zip file here. Then, I can unzip it on my computer and then work with the individual file as needed. So, going back to my slide set here. So for set level workflows, instead of using the sample level data table, which was training_data, we'll need to use a set level workflow here. In this case, you can see training_data_set. And then, just do the other inputs as you would expect, but there are a few variations there. So for the sample level data table, typically if, let's say, we needed to input a column called, read one, for example, we would just do this.read1. For the set level workflows, there is a slight difference in that we take in this dot and then we put in the sample level data table name, but then add an S and then put dot, then the attribute, or rather the column name. So I'll just explain that here. So if I can just open this. Demo flow. Actually, let me give an example before we actually undertake. So just open this as well. So this is our training data, the one that we did genomic characterization. And we said that RC typing does take in assemblies. So our Theiagen does give you an annotated GenBank file. Let's see if it can give us a preview here. Okay, it can't preview the file. So RC typing requires that your input assembly has to be gzipped. But you can see that, our annotated file is only .jbk. So we need to have this as .gbk.gz. And currently, I don't know any workflow that would immediately do this and zip these files. So what I wanted to do was to have all these files zipped into one file, zip file, and then I can be able to download it, unzip it onto my computer, and then gzip using a command line method. So what I would do is I go to the workflow here. And you can see that this column is called proca_gbk. So I would select the training data set. Yeah, not the training data table, because these are set level workflow. And then I would say select table. So if I have already, so there are two ways of doing this. You can literally actually to go back here. I can, let's say, select all the 42 and then remove these that don't have any annotation file. I can save the samples that I have selected as a set. So I can create a set from this sample level data table. And you can see, if I just say that, I can say test set, and I save that. If I go to my training data underscore set, so the set level data table, you'll see that it has created a set level row here that includes the 40 entities that I selected. Okay, so that's one way you can pre-create that set. The other alternative is I can create a new training set within the workflow. So I don't have to have created it before, but when I just select the training data, the set level data table here, I can select, like, I want to create a new training set. I'll select, in this case, all of them. I can remove those two. And either you can use this auto-generated name, or you can also name it the way you want. So let's say test set 2. And then I click OK. So in this case, you are not creating it first and then coming back to the workflow to select that set and then run the workflow, but rather creating the set at the same time as you're selecting the data and launching the workflow. Okay. So and one thing you'll notice here is when you select the input, so remember our set level data table is called training underscore data underscore set. So for the input, remember again, the column that we wanted to select is the proca underscore gbk. So here we'll say this dot training data. So that is where the our sample level information is. And then we add the S here. So it will be training underscore datas. Okay. And it doesn't matter like what your table is called. You just have to add that S and then put another dot, and then you give it the column name. Okay. So that's what we are describing here. So for a typical sample level data table, you have this dot attribute or the column name. But for the set level workflow, you have this dot sample level data table name, then you add the S at the end, and then dot the column name or the attribute. I know that's a bit confusing, so I'll probably stop for a second to if there are any questions. Any questions?
Questions?
Yes, what
I have to use it before we can upload it on the RC side. The workflow.
I only got of the workflow.
Look outside.
Okay. I'm so sorry, your network is a bit jumpy, but yeah. Um, anyone else as Rotimi is typing? So no, so RC typing is actually set level workflow. Because remember, it's undertaking phylogenetic analysis, so you need to combine a set of samples to undertake phylogenetic analysis. Even though I think, yeah, it seems like because, it has to take each sample and then, select the 16 conserved, but ultimately because, it has to then concatenate for each sample and then do the multiple sequence alignment. So it is actually a set level workflow. Yeah. So I, Rotimi, have not investigated if there is, and probably there is a workflow that can actually gzip within Terra, which is sample level. It's just that at this point, I am not aware, but I do know, like, within the Theiagen ecosystem of workflows, so we have that zip column content, which we, will zip it, and then you can be able to download the whole set of files with one download, and then, you can now process it locally and then like upload again. So that's what I was able to do. Yeah. Okay. For your workspaces, you don't have the RC typing workflow. So, but we'd gone over this. So you could get to the workflows tab. Here on find a workflow, you just select the tile and then go to Docstore. Once you have Docstore open, RC typing, it's only one workflow that I know is called RC typing. So we don't even need to, select the organization. So it's pretty easy to find. And once I do RC typing, no spaces, you can see that, I get the workflow JIDC trust /ribosomal widow RC typing. So if I select the workflow on the right here, it gives you different options to launch the workflow with. We are using Terra, so I'll select Terra. And then it brings you a pop-up where you're supposed to select the destination workspace, which should be your workspace. In this case, for me, I would want it to go to this demo workspace, and then I click import, but I expect it to, complain that, there's already a workflow called RC typing, and it's asking me whether I want to overwrite the workflow. So I'll just say, overwrite, because, it's just the workflow that is being overwritten, so no problem at all. And also it's better this way because, it shows, all the values not input here, so we can do a step-by-step with you. Okay, so what you'll need to do is we need to get the data that we need to run RC typing on. So if you go to the training resources page, we are currently in week 3, which focuses on RC typing. So here you'll see in this toggle button, we have the data set. So there's a link here that takes you to this GitHub page. Oh, this is actually cool. So we are able to actually just download this file. So let me just put it here in my downloads. So if you save that file, let me just open this new Finder window. Is it called again? Ribosomal. Okay. Okay, so let me just put it here because it's easier to manage. So you'll get this zip file, which if you unzip, it should open this master file. Then you want to go into tests and then data and then GenBank. So in this folder or directory called GenBank, you'll see a set of files here, right? So these 10 files need to be uploaded onto your workspace, the same way we uploaded read files. So just upload them onto your workspace, and then, as expected, you'll need a TSV file that accompanies these files. And what data table should we create? So create a data table called, what did I go with here? RC typing tutorial. So you'll need to create an RC typing tutorial data table. In this case, you can see I have my files here. The names should be the accession number without the version. So you can see the accession numbers for the different assemblies here. Again, just without the version, like the .1 here. So once you have the set of files uploaded, and then you've uploaded the accompanying TSV file, which creates the data table, now you're ready to run RC typing. So I'll do this from here. Let me just go to my workflow. So we have RC typing. I want to select the latest version, which is version 112 there. And then run the workflow inputs defined by a data table. So in this case, I'm going to select a different table. So this would be the RC typing tutorial set. And then I'll go to select data. As I said, you can either choose an existing data set which you won't have. So the option is to create a new one. And here you just select all the 10 samples there. Then click OK. And once you select, you can see here is, it's saying one RC typing tutorial set containing 10 RC typing tutorials. So essentially a set that contains 10 samples. Okay, so use call caching, select this option as always, and also select ignore empty output. So just so that you don't have columns that are empty throughout. For this one, we don't expect that to happen, but it's usually just good to get into the habit of just selecting that particular option. Now, for the inputs, you can see that there are 3 required inputs here, which is the GenBank set of files, the proteins, which are the reference files, and then the species that you're running. So at the moment, like I said, this does not actually add anything to the analysis because the WDL implemented version does not cover the whole SNCMEC implementation, original implementation. But the hope is that, this will also be incorporated into the current WDL version, or the one that we are running on Terra. So we start with this dot. We know our training or our data table is called RC typing tutorial. So I'll just select that and then, delete everything else. But then we know we need to add an S to the sample level data table name. So I'll just add S, so tutorials, and then we add another dot. And then we need to give it the name of the column where the GenBank files are. So I'll just flip to this other tab to help us. So for me, what I typically do is open 2 different tabs so that I can just, in this case where it's not auto-completing. Like you remember on the sample level workflows, it was quite easy because it was sort of like auto-completing the column names. But for the set level workflows, this does not happen automatically. So this column is called genbank underscore GBFF. So you can either just copy it or, memorize it. And then I'll just come here and paste it there. So this dot, the table name, adding the S dot, then the attribute, or rather the column name. Now we have the RC proteins. Remember, this is the references that we have. So the reference is from the workspace data element that I showed you earlier. Again, if you, okay, so if you have uploaded your files, I've pre-prepared the TSV file, so you can use this one instead of like creating your own, so that's good. And let me see, on your workspaces, I think I'd already pre-created the workspace data element, so you don't need to do this again. So we do know that our key value is Staphylococcus underscore xylosis. Again, for me to avoid typos, I like just, copying that. And I'll go back to my inputs here. So I just need to type workspace dot, and it's auto-completed this for me. I can just verify that that is what I expect, correct? And then the species here is, I can just say Staphylococcus. But like I mentioned, this is not currently implemented, so you could literally type anything and it should be fine. Again, I'm putting this in quotes because you can see the type of the data input here is string. This is a file and it points to that workspace data element. This is an array, which means it has a collection of files. So we don't need to put any of the optional inputs. And then for the outputs, you remember we said it's good practice to just click here, use defaults, so that all these outputs are populated. Otherwise, if you run the workflow without doing that, then you might end up with the workflow running, but then you have nothing returned to your data table. So once you do that, then you just click save, and then you can launch the workflow, right? So I'll launch the workflow, and the workflow should run. Doesn't take long. And once this runs to completion, you come to the set level workflow here. And in my case, I'd run this before, and you should find a tree and RC type underscore tree file. So again, remember that because this is a set level workflow, the data results is populated on the set level data table instead of the sample level. So if you download this, I have raised this with the dev team so that, this currently the file extension is .log, but it is a tree file. And here I'm going to open something like Fandango. So let me just show you what I will do here. I'm going to download this file. And yes, store it in my documents. Let me just come back here. So it's this file here. You can see it has the extension .log. I'm just going to rename it. Instead of .log, it's a NEWIC file actually, as you can see from here. So I'll just use the .nwk notation. It's going to ask me if I want to change. Typically, I don't think on Windows it does ask, but on Mac it will. Now, once Fandango tree, So we'll talk about Fandango, one of the ways to visualize your tree. So I can just take the tree that we just downloaded. I'll just drop it there. It's a very nice tool. You just drag and drop your files, and you can even add metadata, and you can see the tree that has been generated here from this set of 10 sequences is right here. Are there any questions? No questions? Is that because it's a lot? People are confused? Anyone? No questions at all, so that's good.
So, James,
Yeah.
hi, James.
Yeah, hi.
So on Thursday, we'll be going over the same, trying to like implement or doing an exercise on the RC typing ourselves on Thursday.
Yes, and I was thinking of, yes, we do RC typing using this. So what I would like you to do is try using the data set on the data set that you download from week 3 here. But I've also planned to give you another exercise using the samples that you ran through Theiagen. So I'm going to send you an email to describe what you need to do. Like that should be like a second exercise, but with a bit of a twist, because hopefully, you can be able to remember last week we talked about QC. So what I'll want you to do is select the samples that you think. Remember, we ran 42 samples, 2 we expected to fail, so 40 should have passed. And then out of the 40, not all of them pass QC. And remember, QC is a broad way of saying, like, are they the right pathogen? Are they the right assembly length? Are they the right, GC content, and those sorts of things? And then you select the samples that you think pass QC. Again, remember that, this genomic characterization. So for Vibrio, we had O1, 39. We have ELTO, we have, the classical type. So, eventually, I want to see like the thought process, like, okay, which samples do you pick for your phylogenetic analysis? Again, remember, we said samples should be similar to each other. So for example, they should be the same ST and things like that. And then once you select this set of samples, what I'm going to do is give you already gzipped files for all the 40. And then now it will be up to you to select which of those you think passed QC, and then you can run them through the RC typing workflow.
Okay.
So, yeah.
Okay, we can try doing that. Thanks, James.
All right. Thank you all. I think we've come to the end of the session today. So I'll follow up with an email. Again, you should be able to find the recording for this session under the recording section here shortly. You can go over the slides as well, which should be under this, will be under the slides here in a few minutes. And then if you have any question, of course, just do reach out. Hi, Rotimi.
You missed my question. I typed it on the chat box.
Whether we might use this for speciation. Well, of course, because this is conserved across, different pathogens. So I would expect that, yes, you would use them for undertaking speciation analysis. I don't see why you wouldn't. So I think, yes, you can use it to undertake speciation, unless, is Claire still here? She could say otherwise. No. Yeah, but I think principally you should be able to undertake speciation analysis with this. Okay. I think we'll end it there. Thank you all and see you on Thursday.
Thank you, James.